So I'm going to talk about speciation in bacteria and beyond, characterizing evolutionary patterns of diversification. And this is work with various people who I'm crediting down here. So the general question I'm interested in is, what are species? Does life really evolve into these simple units? Or are species more of a convenient summary of complex diversity patterns? And in particular, how do patterns of diversity vary among groups? Can we identify common features in very different organisms? So bacterial species are interesting from this point of view. Firstly, they have very varied mechanisms of gene exchange. So they engage in clonal reproduction, but also in exchanging genes through homologous recombination and various other mechanisms. Of course, they're an extremely diverse group, so statistically speaking, all organisms are bacteria, so we should pay some attention to them then. But they have this lack of traditional phenotypic characters, so until recently, patterns of their diversity were very hard to look at. So partly because theories of speciation have been developed mainly for eukaryotes, sexually reproducing organisms, then there's been this widespread view that bacteria don't have species. But recently people have looked at the evolutionary mechanisms that might act in bacteria and identified that the same processes that we talk about for speciation in eukaryotes, and they, in principle, they can apply. So first of all, you've got these kind of ideas, something like reproductive isolation, where you might have groups where recombination can occur within those groups but not between them, or exchange of genes. And these have been developed particularly for disease organisms and studies of multilocus variation. And then secondly, divergent selection can cause divergence between different groups, and these have been particularly popular for environmental studies and looking at the ecotype definition of species. But these things haven't really been put together to explore what's going on in a particular clade. So the very general question then is, do evolutionary forces in bacteria act to generate something approximating species where you have these simple, relatively simple units of diversity, or is a more complex model of diversity required where some genes are jumping around, some aren't to the same extent, and so on? So we set out to test these ideas using this particular group, so the Bacillus cereus species complex. So these are found widely in soil and on plants. They're very easy to isolate members of this group. There's been quite a lot of interest in them in terms of causing foodborne illness and as biocontrol agents, which means there's a reasonable amount of information there already. And in particular, there's been development of a multilocus sequence type scheme. So this is a common approach in bacteria for surveying diversity, is to use a suite of maybe 7 genes to look at diversity. And from surveys across bacteria, then they have intermediate levels of recombination, so they fall in quite an interesting point in parameter space for understanding what the patterns of diversity might be. And then for those of you who like your study organisms to be cute and cuddly, then what could be cuter than that? So, the data I'm going to show you, a sample, is this group, and we sampled them from a 100 by 100 meter plot at Silwood Park, which is the campus that I'm based in. My office is sort of there somewhere. And we surveyed for a few hundred isolates that we collected. Then we surveyed the standard housekeeping genes, the ones that are normally used for multilocus studies of this group. So there's 7 of these genes here, and they're used for basic kind of housekeeping processes within the cell. So the expectation is that variation in them should be effectively neutral, because they're always performing the same job in each of the different isolates. And then we're aiming to sample 7 so-called ecological genes as well. At the moment we've just got 3 done, so I'm going to tell you about those. So these are genes that actually do something more interesting, and where we think that they're likely to be targets of divergent selection among the different isolates. And I'll explain more about them later. So there's 2 sets of analyses then to look for evidence of these different forces that might be generating diversity in this group. So first of all, to test directly for arenas of recombination. So groups where you have high rates of recombination within the group, but then low rates between them and another group. And the approach here is to use a model to optimize the membership for those groups. And then secondly, to look for arenas of divergent selection. So to find groups where there's very little divergence in amino acid sequence in the ecological genes within the groups, but then there's a high divergence in amino acid sequence and potentially function between such groups. Okay, so first of all, we're using this infinite alleles model that was developed by this group here. And this was developed for a single bacterial species to explain the pattern of multilocus variation based on fitting parameters of population mutation rate and recombination rate. So the data that goes in here is the number of allelic differences between pairs of isolates that you compare, and then through fitting this model you get an estimate of recombination rate and mutation rate. So the change that we've made to this is to start by fitting a single model to the whole tree. So this is kind of norm model that there's been no diversification into independently recombining groups here. And then to successively, so you can calculate a likelihood score for that, and then to successively split the tree into separate groups where you fit a different infinite alleles model to each of those groups. So you're saying, and then you assume that there's no recombination between those groups. So you have a different model for the within group comparisons in the two groups separately, and then a model for the between group variation that assumes there's no recombination. And I'll skip through this for the basis of time, but you keep going down. If, at some point, if you try to split the group further, but the likelihood score actually gets worse, then you reject that, and then you can try the other side of the tree. So we're trying to optimize groups that are independently recombining from one another. So this is showing the housekeeping gene phylogeny now for all of the isolates from our sample. And then if you perform the algorithm on here, then you split this into these separate groups. So in particular, there's a major red clade here, orange clade here, and green clade here, where the model suggests that there's recombination within these groups, but not between them. And that's just to show the details of how the model fit proceeds. So there's significant evidence to rule out the null model that everything's just one recombining whole. Okay, so the next part then was to look for arenas of divergent selection. So first of all, we could ask, well, do the ecological genes fit within these groups that we've identified for recombination based on just a housekeeping gene variation? And secondly, can we see any evidence that there's groups that are experiencing divergent selection on these genes? So this is the first one we're looking at. It's this HBLA gene, and so this is a toxin that these bacteria produce. It has some nasty effects in humans, but it's involved in them kind of entering into some of these habitats that they're founding. If you survey very generally what's going on here, then you can fit this model where there's a percentage of sites, fairly small, but a significant percentage of sites that do appear to be under positive or divergent selection. So then we need to look at how this relates to the grouping that we found from the housekeeping genes. So this is the housekeeping gene tree, again, with the colors showing the groups that we've identified as being independently recombining from one another. And now this is the tree for the ecological gene, and you can see that there's some kind of broad structure that these groups are still found. So these three major groups, you've got mainly red isolates down here, mainly green here, and sort of predominantly orange up there. So there's some broad structure that's the same, but you can also see that there's been some exchange of genes between these different groups. So there's some groups that are actually going in the blue group, in the blue clade based on housekeeping genes that actually have a copy that's more like the orange copy for this ecological gene. And then now I'm showing the trees, but only resolving branches where there's been a, where the change in amino acid relative to silent changes in these genes is greater than 1. So these are evidence of branches where there's been positive selection or divergent selection between groups, if you like. And you can see in the housekeeping genes there's not much going on. These are probably artifacts of just dividing small numbers. But in the toxin gene, then these groups, the green group especially here, the red one, then they are separated by evidence of significant divergent selection, although there's additional groups within the red group down here. So for this gene then, there's the same broad structure as the housekeeping genes, but some recent transfer, and we can find evidence of groups that have been experiencing divergent selection. The second gene shows more or less the same pattern as that one, so I won't talk about that further. This is a gene that's involved in kind of communication between different cells and switching on some of these toxin genes. And the final one is this citrate sensor protein that regulates the ability to use citrate as a carbon source. Again, there's evidence that part of this gene is under positive or divergent selection. And in this case, the pattern's much more complicated. So you can still sort of make out a green group up here, but the orange and the red groups are much more mixed together. So it's not obvious that the same structure is really being found here. And in particular, there's a very striking pattern of divergent selection, that you've got loads of different variants that differ greatly from one another in amino acid sequence. And so there's kind of extreme levels of divergent selection that seem to be acting fairly independently from the housekeeping gene groups that we found. So in terms of answering the questions then, do the ecological genes fit with the housekeeping genes? These two sort of do, although there's evidence for some exchange among the different groups. But in the case of the final gene, this CYT-S gene, then really it doesn't. It seems to be doing its own thing. Can we identify groups that are experiencing divergent selection? Yes, we can. So this is just showing it again in another way. So this is a kind of formal way that you would be able to test for the existence of ecotypes, say, although the answer you get might differ depending on which genes you look at. So to conclude then, there's various interesting features about this group in terms of there's an intermediate recombination rate, but then at the same time, as with other bacterial groups, there is this possibility of exchanging DNA between more distant relatives as well. The outcome of this seems to be that you can identify something like reproductively isolated groups if you're looking at these boring housekeeping genes, but potentially you get a more complex pattern of diversity if you look at ecological genes. So a more general conclusion, the same processes are operating as do in sexual eukaryotes in terms of divergent selection and origin of isolation, but the outcome is a bit different because of these unique features I mentioned. Is a true pattern of diversity more complicated than simple species? Yes, it does seem to be in this case. Are species still a useful concept? Well, probably they are, because the housekeeping genes are still showing some kind of congruence. So at some level, there's a pattern of diversity that you could summarize in terms of simple units. And then I'll leave you with thinking about sexual eukaryotes as well. So this kind of interrogation of the pattern of diversity within a clade and where the units fall depending on different processes that might be acting on them is something that hasn't really been done very much in sexual eukaryotes either. And so possibly some of the complexities that we're finding here might apply in sexual eukaryotes as well if you did this kind of thing. Okay, thank you very much.
Thanks a lot for an exciting talk and for bringing us back on schedule. So, some questions? Question was also
very short remark about it. But you say bacteria doesn't have species. You must say it doesn't have biological species.
I was quoting other people when I said that.
Biological species is one thing and species is another thing. If you apply that criteria, are you saying that those are, you can find biological species in bacteria, or that bacteria show kind of diversification that can equate to concept of species, which concept of species can be what they, the last talk call the concept of cohesive species, put forward by Alan Templeton many years ago. I think that this was misregarded by many evolutionists. What do you think?
Yes, I agree. I mean, I sort of skipped over it a bit, but I did say that because the concept of species was developed mainly in sexual eukaryotes, that people had said bacteria don't have species, and you're right that if you apply strict biological definition, then it can be difficult, especially in the groups where there's very low recombination. But even in groups where there's no recombination, there are still forces that could cause cohesiveness, like you say, and produce a pattern that's equivalent to species.
Because as you know, E. coli, for instance, has about maybe 18% of genes which that don't belong to E. coli, but still is E. coli, you know. How do you explain it?
Yeah, I agree.
Okay, other questions or comments? Mike?
That was great and not really confused. The question I have is if you can think of an alternative explanation besides reproductive isolation to explain the patterns you see. And the reason I ask the question is that it's very hard for me to wrap my head around the notion that the bacteria are in fact segregating reproductively for some genes, but yet others are not segregating for, and even segregating in different ways. And I'm just wondering whether there's not an alternative hypothesis that you could generate, or maybe 2 or 3 alternative hypotheses.
That raises a lot of interesting questions. I mean, I think the question is whether the different extent to which these genes are moving around is because of some kind of mechanisms that determine that some are more likely to be taken up, or whether it's more the outcome of that there's reasons why it's beneficial for these ecological genes to be taken up. Therefore, those are the survivors that you see, and there's more evidence that they're moving around.
Or that the housekeeping genes, perhaps, are bound together in some kind of integrated relationship.
Yeah, I mean, it's not a physical relationship on the genome, because they tend to pick these housekeeping genes to be dotted all around. But I think you're right, that would be something very interesting to look at. I mean, the other thing is, it's possible that rather than there being strict groups, that it's more like a decay with distance, and that's something that, using the same model, you could test. That we haven't done that. So is it really that there's discrete groups where they're kind of switching on and off, whether they recombine or not, or is it just a decay with the amount of divergence over time?